Hello and welcome to my pre-recorded talk. Some of the best understood hormone behavior relationships are between testosterone and social behaviors, including aggression, courtship, and parental care. And there are multiple ways that testosterone can influence the different aspects of social behavior. Testosterone can act through multiple biochemical pathways. For example, it might bind directly to androgen receptor or be converted to dihydrotestosterone, which more potently binds to androgen receptor. Or it might be converted to estradiol and act through estrogen signaling pathways. And these actions can occur through the multiple nodes of the social behavior network in the brain, and thus producing multifarious ways that testosterone can regulate these different behaviors. So the white-tailed manakins have an unusual social behavior, a cooperative lekking display, where territory-holding males will perform with subordinate or floater males. This cooperative dance is mutually beneficial, as more cooperative territory holders are more likely to gain paternity, and more cooperative floaters are more likely to gain a territory later in life. Recent work has shown that testosterone modulates social behavior differently depending on whether the bird is territorial or floater. Here in red, we see that as testosterone increases in floater males, they become more social, but the converse is true in territorial males becoming less social. And territorial males also, on average, have higher testosterone than floaters. And to delve more deeply into these results, see the talks given by the fabulous Ros Dakin. So we were interested in looking at what genes and gene networks might be involved in this unusual male cooperative behavior. And specifically, we're seeking to answer what genes and neuroendocrine pathways modulate individual variation in cooperative behavior and testosterone concentration. We're also interested in understanding what genes and neuroendocrine pathways might be involved in the status-specific regulation of cooperative behavior. And then finally, we want to know how these genes and pathways are associated with behavior, both within and between specific neuroendocrine tissues in the social behavior network and beyond. So to try and answer these questions, we sampled 12 tissues across the social behavior network and the HPG axis from 12 individuals. Each tissue and each individual is a unique RNA sequencing library. Now, the work here is an extension on the molecular and cryosectioning methods shown in the talk by the wonderful Brent Horton on Thursday, so go there for more details. So going back to our questions, how does gene expression vary within and across the social behavior network according to individual variation in hormonal and behavioral traits? So we predict that we will see different patterns of gene expression across the brain in relation to our different traits of interest. And this prediction is based off previous work that shows differential activations of neurons in the social behavior network relating to different aspects of social behavior. And so down here at the bottom, we are showing the different nodes of the social behavior network and the different activation indicated by the peak height, the different behaviors. And so therefore we expect a similar landscape of gene expression across the social behavior network, with different gene regulatory networks involved in regulating social behavior. So these nodes of the social behavior network are defined by different densities of different hormone receptors, steroid-sensitive neuropeptides, and steroidogenic enzymes. And so based on these known associations and long neuroethology work, we have selected a set of genes that we expect will be responding to testosterone and involved in modulating social behavior in the wire-tailed manakin. But today I'll just be focusing on some genes that interacted directly with testosterone: the androgen receptor, aromatase, and reductase. One way we're looking at variation in gene expression within and between nodes of the social behavior network is through weighted gene co-expression network analysis, or WGCNA. This method takes correlation coefficients between all pairs of genes and turns these into a network of relationships between genes. These networks can then be divided into coherent subnetworks of genes that have similar expression patterns. These subnetworks are called modules and are assigned a color at random, like this bar above. These modules can then in turn be correlated with the traits of interest, which are the rows, And the colored squares indicate the strength and direction of the correlation between the module eigengene and the trait. And so red colors indicate a positive correlation between gene expression and the trait, and blue indicates a negative correlation or downregulation according to the trait of interest. So this figure actually comes from the medial preoptic area and is rich in information. And so what we can see here is that different sets of genes are associated with each of our different traits: testosterone, time spent at the lek, and sociality. And in particular, this brown module here is highly positively correlated with testosterone, where 237 genes are regulated in a similar fashion. And genes with a high module membership are putative drivers of gene expression in that module. And so our top gene in this module by module membership is androgen receptor, which is known to bind to testosterone and act as a transcription factor regulating sweets of genes downstream. And so here as testosterone increases in the preoptic area, sensitivity to the testosterone signal also increases. So then if we zoom out to two other nodes, the nucleus taenia and their lateral septum, we have different patterns of expression. Each of these figures were generated independently, so a blue module in one isn't necessarily equivalent to a blue module in the other. But what we can see here again is this recapitulation of different gene modules associated with different traits. And what we can also see is that there is variation across the nodes in the strength and the significance of association with traits in a gene expression landscape. So for example, there are fewer modules with high correlation in, according to testosterone in the lateral septum, potentially indicating that it is less involved in response to testosterone. Okay, so I've shown you a snapshot of some of this individual variation, but how can gene expression modulate status-specific responses to testosterone across the network? Do we see differences in average gene expression according to territorial or floater? Or do we see genes regulated with testosterone in a status-specific manner? So these are the results from differential expression analysis across all of our 12 tissues, which is shown along the x-axis. The y-axis represents the log-fold change in gene expression, where negative values indicate lower expression in the territorial males relative to the floater, and positive values indicate higher expression in the territorial males. The circles in the plot are color-coded according to each of the three candidate genes that interact directly with testosterone: the androgen receptor, aromatase, and reductase. Circle size indicates the average expression of that gene. So as you can see here, there is also a landscape in the absolute expression of our three genes across the social behavior network, as indicated by the circle size, as well as how much expression changes according to the social status, as indicated by the position of the circle. And then finally, the filled circles indicate genes that are significantly differentially expressed prior to FGR correction. So these genes can also be plotted like this. So here we have two nuclei, the preoptic area and the bed nucleus of the stria terminalis, that appear to be more sensitive to testosterone in territory holders, and the nucleus taenia appears to be less sensitive to the testosterone signal in territorial males. In the lateral septum in territorial males, we have higher expression of 5-alpha reductase, which converts testosterone to dihydrotestosterone and binds more potently to the androgen receptor, and thus providing a potential mechanism for this status-specific regulation of social behavior. So not only do we find differences in average expression, but we also find an interaction effect between social status and testosterone in a number of tissues that are highlighted by these stars. So in these 4 nuclei, we see status-specific effects of testosterone on gene expression. The top 2 panels show androgen receptor has a positive relationship with testosterone in floaters. And perhaps in the lateral septum, this is reducing sensitivity to the more potent dihydrotestosterone, which appears to be converting at a higher rate in the territorial birds, as shown by a previous analysis. And so maybe in the lateral septum, there's no net effect in status-specific regulation based on these results. And so we can also see a status-specific relationship in the nucleus taenia for aromatase. And this gene converts testosterone to estradiol. And so is potentially, the estrogen receptors are involved in behavioral modulation in the nucleus taenia. So what these last results show is variation across the social behavior network in genes known to influence testosterone's action in the brain. And these differences in expression may underlie status-specific relationship between circulating testosterone and behavior. We also see more broadly that we have putative gene regulatory networks and known candidate genes associated with both social status and individual variation in endocrine and behavioral traits, and that these show variation across the social behavior network in a gene expression landscape. And the final exciting thing about this work is that it's the first study of social behavior using a system-wide approach in a wild bird, showing the potential of these methods to look at mechanisms underlying complex behavior expressed in natural environments. And we are only just scratching the surface here. So I want to thank my PIPRA peeps, my collaborators. They were responsible for the inception of this project, as well as the execution of fieldwork, endocrine work, and the lab work. Thank you for your hard work in mentoring me on this project. Are there any questions?